guys, it's Mrs. Gary. I hope you're doing well today. I realized that I did not tell you about the 3 different types of RNA when I gave you your video the other day. So I'm going to talk about just briefly 3 different types of RNA. However, for this week, you only need to know about one of them. So the first one is about messenger RNA, and that's this first one here. M stands for messenger, and basically what we're going to be doing, as I told you before, DNA is too large to leave the nucleus. And so what we have to do is we have to make it into a smaller molecule. And if you remember, mRNA or RNA is just single-stranded, so it's just half of that double helix. And so it is small enough to be able to leave the nucleus and head to the ribosome where proteins are then made. And so when we transcribe DNA into messenger RNA, basically what that means is we're decoding the DNA into RNA so that it can be brought to the ribosome to make a protein. The other type of RNA that you'll be coming across is called tRNA, and that T stands for transfer. Its job is to bring amino acids into the ribosomes. And if you remember that amino acids are what makes up proteins. And so when we learn about translation, we're going to hear more about tRNA. And these tRNAs are found in the cytoplasm. And then finally, the third type of RNA is called ribosomal RNA or rRNA. This is basically just part of what the ribosome is made of, and it helps to move the mRNA along in the ribosome in order for more proteins to be made. But we'll talk more about that later. So the reason that we're talking about all of these again is for this process called protein synthesis. And so when we're talking about protein synthesis, we're talking about DNA, and then making that into a code that can leave the nucleus called RNA. And then finally, that RNA is going to head to the ribosome where proteins are made. And if you recall, proteins make everything about us, from the color of our hair to the color of our eyes to how tall we are and all of the other things that go on in our bodies. And so that is the purpose of this lesson. And so let's talk about transcription. So transcription is a process in which RNA is made from DNA. Like I said before, DNA is too large of a molecule to leave the nucleus. And so what has to happen is we have to make it into a smaller molecule. And again, RNA is a single-stranded molecule, and so it's going to be able to fit outside of those nuclear pores and head into the cytoplasm towards the ribosome. And so the picture I'm showing you right now is showing you how that is made. And so basically what happens is the DNA unwinds, and then we have RNA polymerase is going to come through, and it's going to make a messenger RNA strand, which is this red strand here. It's going to match up those bases, those nitrogenous bases, the A's, the U's, the C's, and the G's, and those are going to match up with your A's, your T's, your G's, and your C's on the DNA. And so that is the whole purpose of transcription. So first thing that happens during transcription is that DNA is unzipped by helicase, just like when we are replicating DNA. So you can see in this diagram here that that blue molecule right here, is what's going to break those hydrogen bonds between the base pairs and separate or unzip that DNA molecule. The second step is the other enzyme, RNA polymerase, and what it's going to do is it is going to add the RNA nucleotides to the DNA strand. And so it's going to add our A's to our T's, our U's to our A's, our C's to our G's, and our G's to our C's. And if you remember, I mentioned the other day that there are no T's or thymines in RNA. Instead, we use the letter U, which stands for uracil. So the third step of transcription is when messenger RNA ends up being complete when the RNA polymerase reaches a stop code in the DNA. And we'll learn more about that when we get to translation. But basically, there's going to be a sequence in the DNA that tells the RNA polymerase to stop transcribing that DNA into RNA. And so then once the messenger RNA is made, it's able to leave the nucleus and carry that DNA code to the cytoplasm, eventually going to the ribosome where those proteins will be made. So I want to make sure that you guys understand how replication is different from transcription. So if you look here, we have our A's always match our T's, and our C's match our G's in DNA replication, because we're only talking about DNA. And our nucleotides only contain the nitrogenous bases A, T, C, and G. But the thing that's different in transcription, remember, is we're dealing with RNA. And RNA does not have thymine or the T's in there. Instead, wherever we see an A on the DNA, we're going to match it with a U from the RNA. But the C's and the G's are going to stay exactly the same. But then if you look down here, we see a T in the DNA. Well, T's are always matched with A's, and there's A's in RNA, so we're always going to match those together. It's just whenever you see an A in the DNA and you're transcribing it into RNA, we put a U there instead. And we will be using the other half of our model to model transcription once we get back from break. And so just keep in mind that the original DNA strand is this is this blue strand here, and the other strand that is made, the daughter strand in DNA, is going to be this red one over here, and this red and yellow one over here is going to be our messenger RNA. So what I would like you to do is just pause the video for a second, and I'd like you to transcribe this following sequence and see if you can figure out what the messenger RNA is going to be. So I'm going to pause the video for just a second and give you a chance to do that. Okay, so hopefully you had a minute to put your answers onto your paper, because this is a really important process that you need to know, because you're going to have a mastery project that you're going to be doing in a couple weeks, and you have to get really good at turning DNA into messenger RNA. And so let's just take a quick look and see if you got the answers correct. So let's take a look and see. So as you can see here, if you want to check your answers really quickly, so wherever I see a T on DNA, I'm going to put an A for RNA. And whenever I see an A on DNA, I'm going to put a U for RNA. So please remember, it's really, really important for you to remember that there are no T's in RNA. So whenever you're making any kind of RNA, you only use U's instead of T's. So every time you see an A in DNA, you're going to match a U for RNA. So if you look at all of these A's, you're going to see that they all match up with a U. But do remember that wherever you see a T in DNA, it's still going to match up with A on RNA, because there is still A's in RNA. Okay? So then what I would like you to do next is, I'm going to back out of this PowerPoint for you and show you your document that you have to use for your homework. And so what you guys are going to do is you guys are going to practice transcribing DNA into messenger RNA. And I've already started this one for you. So if we look at the directions, it says transcribe the following DNA sequences into messenger RNA transcripts. So that's mRNA. If you double click on each DNA gene, it'll open Google Draw, just like that. And then all you need to do is to add in your matching mRNA letters. So I've already done the first three for you. So where A is, we put a U. Where T is, we put an A, and for C, we put a G. So if you were to look at the next one, think about what that letter is going to be, okay? If you have any questions or are completely confused, please, please, please use my office hours. Come on anytime to the link that I've given you on Google Meet between 11 and 11:30 each weekday. And please go on and ask questions, and I can give you some more practice, or I can help you out with learning this. But keep in mind, you've got to be really good at this, because the next thing we're going to do is we're going to be learning about translation. And that's going to be a little bit more complicated. So please, please, please ask questions. I'm available on other times, not just during office hours, so email me with questions. But please, please, please contact me if you need help, okay? This is probably the last you'll hear from me before break next week. You are not expected to do anything over break. I just want you to relax, have some fun, get some air, take your pets for a walk, go get some exercise. Don't stay inside all day. The weather is too beautiful for us to stay inside all day. Okay? So please do your best to relax and catch up on any work you need to do. But have a great spring break, and I will see you back when break is over. Have a great day, guys. Miss you all. Bye.